Dr. Tissot. Now we move on to next speaker, who is Professor Giacinto Scoles from Temple University, Philadelphia. He will be talking about a new nano workbench to study and control the formation of oligo fibrils made by Alfa Synuclein in Parkinson's disease.
Sorry. Thank you. Okay. Thank you. Thank you. I'm here today in a double role, as a patient and as a scientist. I have Parkinson's disease, and I'm fed up with it, so I decided to do something about it and start investigating it. 5 years ago, I wrote a grant for the ERC, and I was not yet pessimistic about my Parkinson. I had only micrography, a few other symptoms like this. And then I wrote a grant on cancer. We did some progress on cancer. The grant finishes on Monday, July 1st, or July 4th actually. July 1st finishes the grant. We don't let you spend a cent outside of the team that you wrote a grant on. So I decided to start changing my field of research and start with Parkinson's disease. And I will tell you why in a moment. I like to make a workbench for making fibrils. The fibrils are more complex than what I can make with Alfa Synuclein alone. But with Alfa Synuclein, you can form already some fibrils. And I will start with those. Actually, dissolving those, disassembling those will be easier than disassembling the total complexes that are now forming the substance nigra, but I will start with those. I have good collaborators. Ubako will provide me with mutants of diseases of Alfa Synuclein. DeMarco will go for the nanobodies, Lyle, Sissa, the theory, and so on and so forth. As I told you, I changed the paradigm to study applied physics to medicine. Typically, my colleagues do study material science. They come up with an acute application of material science. It's a good solution. They are looking for problems. They may find, they may not find the problem that fits their applications. I decided to, from the very beginning, to look for a problem, then started taking all the techniques which I needed to solve a problem. And we did that for the liquid biopsy for cancer, which we came up with a solution to look for liquid biopsies, cancerous cells in the blood, based on metabolism. If the cell is altered metabolism, the Warburg effect tells us it is cancerous, and we dissolve the cells in a system of droplets, making an artificial emulsion. We look for acidity of the droplets, each one by one, and if the droplet is acidic, contain a cancer cell. This allowed us to make a machine. We are now gearing up to it's patented and to sell it eventually next year. But now, as I told you, I would like to change field. And I try to understand and control Lewis bodies because it is not even known at present if the Alfa Synuclein, the protein which has a beautiful phase transition, I could not have a better introduction than the three talks that preceded me. Because I learned a lot from them. I'm far from being a neurologist, and not only doing nanomedicine for 5 years in another field, not even neurology. But apparently the consensus is not even known if Alfa Synuclein is the consequence or the origin of the Parkinson. I would say most likely the origin, because the consequences, maybe only the consequences, we'll see. One is to do a prepare a concoction, give it to the brain, and dissolve these fibrils. If the Parkinson remains, therefore, you have a consequence. If the Parkinson disappears, then you have the Alfa Synuclein problems were the origin of the disease. So let's see what happens. Also blepharospasm. My eye goes shut for that time. I have to touch them and open them up. Alpha synuclein is a mostly non-structured protein. It has 140 residues. Some of them are divided in two domains, helical domains at the top, joined by a flexible joint, and then there is an in-between hydrophobic domain. The hydrophobic domain is essential for preparing these fibrils, but then there is a tail, long tail, totally unstructured, which has no apparent function other than complicating things to people who study them. I beg you to read my slides so I don't need to talk clearly about them. 5 years ago, actually 4 years ago, we did already an attempt in a laboratory next to mine, the laboratory of Loredana Casalis. Used to be my laboratory many years ago, then became hers when I moved to cancer studies. And she, with a student, Stefania Corvaglia, did an attempt to find this. There used to be a clock there to monitor the time. It's not there anymore. They tried to graft nanos. I will teach you in a moment what means nanografting. So nanograft Alfa Synuclein on the surface, a flat surface for reference, and you can use atomic force microscope to see what grows on it. And then they not succeeded because it was impossible. It turned out to be impossible to graft the Alfa Synuclein. But then they solved another problem. They published a paper 2 years ago, atomic force microscopy based nanosay, new method to study Alfa Synuclein dopamine bioaffinity interactions, and also interaction with the antibodies. I will analyze this paper in some detail because it will allow me to design a better setup to do Alfa Synuclein studies in a moment. What they tried was this. As you know, sulfur and gold like each other, and there are several more layers of stuff where, oops, I'm sorry. Turn me around. Turn me around. Okay. Okay, like this. Okay. Before. Okay. Fine. Thank you. That's fine. Yeah. Yes. There is sulfur here, and these are hydrocarbons with PEG at the top so that they can form a surface which is totally repellent to proteins, because you don't want to have any protein absorption there. When you use a height to determine what happens on the surface, you don't want any spurious signal due to physical absorption. And this is the nanografted Alfa Synuclein, how it was planned, and this is another part of the surface monolayer. In order to have sulfur here, where the Alfa Synuclein would be bound to the gold, you needed to have cysteine. There were 2 cysteine tagged at the bottom of this protein, and that was it, modified, produced by use of cloning. That was it. This is a surface monolayer of hydrocarbons instead. Image properly with an atomic force microscope. Every 3 gold atoms, there is 1 sulfur atom. The structure is not exactly as this. It's a very old slide, but there is some sort of dimerization between the sulfur atoms, which took us 15 years to solve with X-rays and other techniques, which were able to solve. But basically, the monolayer remains hydrocarbon tilted 30 degrees, which are compact and very much isolating the surface of the gold from the rest of the world. This is that first nanografting. It was invented by Cangiulio in 1997. Cangiulio, former student of mine, now she's professor at UC Davis. You have a tip of atomic force microscope, tip staying out of the monolayer, and you change the monolayer. You put a longer monolayer in, or any type of monolayer in the solution, and dip the tip into the organic monolayer, disturbing the bottom. It generates exchange of the hydrogen, which is here. Hydrogen here, because these are thiols. These are thiolates already. You give an hydrogen to this guy, this guy leaves, and instead a longer guy stays down. Because there are lots of longer guys, very few of these go in solution, they never come back, they generate a too diluted solution. Therefore, then when you're finished grafting the stuff, you leave it with the tip, and then you have done the substitution here. And then you reimagine the surface, and you see the difference. I will show you an example in a moment. This is nanografting C18 with the C8 or C10 with the OH group. The blue groups are OH groupers. We did a paper a long time ago studying this influence of the OH group at top on the homogeneity of this. Try to study membranes and things of this sort. Important point, we did this by nanografting. Show you the results. I found this old slide. This is a C18 into C11OH. As you can see here, the height difference is between 1 or so and 12 nanometers, 11 nanometers or so. The length of this, the difference in length is exactly that between the two molecules. So we know what's going on, and we have improved the techniques tremendously, as you will see in a moment. But this is one of the historical slides in my laboratory where people believed what was happening. You change the long molecule for the short ones. The short one is still there. The long molecule form an island there. The attempt of Corvaglia was to nanograph Alfa Synuclein. She did not succeed, however. Alfa Synuclein was not staying there. The surface, you imagine Alfa Synuclein, large molecules, 140 residues, it would have sticking out of the monolayer of polyethylene glycol and be visible. Instead, it was not visible. Why? Being on such a molecule, and very few of them being able to be grafted, the molecule will be here, will be sitting out of this hole and then wandering on the surface. The tip of the microscope will move it around and never detect it. But if you throw an antibody on it, the antibody sticks to a few molecules which are in these few microns, and then the antibody much larger will form a layer of molecules which is easily visible by white stuff over the black background, which means difference in height of a few nanometers, the antibody is. These, they depicted here wrongly. The alpha synuclein all collapse into this, but it's not possible. The hole will be too small for a molecule like this. The molecule can stick out and wander around the surface and be moved away the tip of the microscope, still not be visible. Okay? But they also made a check. There was not any physical absorption on it by taking the layer of polyethylene glycol, nano shaving as they call it, making a hole, making not with the tip of the microscope, pushing down into the organics, cleaning up there. Since the solution will contain thiols, we'll take the thiols and put it in the solution and make a hole in this. The hole resulted to be as high as it should have been, the height 2 nanometers or so, which is the height of the thiol with a PAG polyethylene glycol on top. That was it. No absorption, nor in the gold, nor in the monolayer next to it. So you can proceed. And this is the experiment they did. They first grafted Alfa Synuclein, then they saw the antibody on it. They detected this saturation curve, which gives them the attraction of the monolayer. Particularly after increasing the DA binding, about 500 nanometers, you can read it there. It was 1.7 micromolar dissociation constant, which is not strong binding, mind you, sufficient binding to start doing something with it. Indeed, it's been known that dopamine can attach to alpha synuclein pretty strongly, and it will displace the antibody. Therefore, they did that. And this time, for a decrease of the height, of course, because the antibody generates this height on top of the alpha synuclein. If you displace it with the dopamine, then decrease the height. You can plot the function of the concentration of the dopamine. You have a nice curve here. Sorry. A nice curve here, both on sigmoid curve, and you find that it's 10 minus 6, 10 minus 4, 10 minus 5, 10 micromolar concentration, more or less, in between here, which is, again, the first time it was determined quantitatively the interaction with dopamine. But the dopamine would have been enough for medicine to dissolve the fibrils. I would have dissolved them in my brain also long ago because I take dopamine every day, about a gram or so. Therefore, I'm not for sure that dopamine is not enough to be. What we are looking to do now is different. However, here is quite clearly dopamine will displace the antibody, because with the antibody, the height of the surface was like this, and then with the dopamine it became like this. So it lost all of this height, which is basically the dopamine height, and again went down to this when the whole thing left in this area region. Yes, I will finish a moment. Now that we have understood why only a few molecules of alpha synuclein have been deposited on the surface, actually, I forgot to tell you, the reason was that alpha synuclein is a linear molecule, diffuses very slowly. Therefore, when the tip goes through the nanografting procedure, the first molecule is around there, graft. The second molecule doesn't come from the solution enough in time before the tip is over. We did quantitative studies of that, and it checks. So we have to go to do the grafting in a different way. We'll graft DNA. DNA grafts very beautifully. We'll graft DNA, make these towers of DNA there. Here. And then variable densities. If we go one time, half a time, the tip is 10 nanometers broad. If every 10 nanometers we do it 10, 20 nanometers, we skip 10 nanometers to another grafting, we have certain density. But if we pass 2 or 3 times, we can put more and more alpha synuclein in there. No, I mean DNA. When the DNA is there, and we know also how much DNA we put down, because DNA will a lot help each other to stand up. And I should have the voice of Barnold's, nice big voice, and I lost it. But in any case, when there's enough density of DNA, we can judge by the height of these towers here. And then we put alpha synuclein in it with the conjugated DNA at the bottom to absorb here, to recombine. And then we can leave overnight, even if it's necessary. DNA will recombine. We basically do what DDI is, DNA detected immobilization proteins. I know a bit over time, but there is 15 minutes discussion which I never taken up by anybody. So I will finish in a moment. After we will have this alpha synuclein oligos immobilized on the surface, we put more alpha synuclein in solution without DNA this time, put some spermidine or amino groups which are well known to favor formation of the fibrils, form the fibrils, detect them by height, and try to dissolve them. I will try to dissolve them. Sarah Fortuna gave a talk yesterday in which she used nanobodies, or talented nanobodies, to produce very lot of interactions between them and the proteins. And we hope with these guys to dissolve the fibrils. If we succeed, we'll take a doses without having FDA approval. I will do myself. Thank you very much.
All right. Thank you, Professor Scoles. I would like to, in view of time, there's not so much time to, to invite all the speakers to come in front. If you have questions to any speaker, just you can ask, because 2:45 you have to move to the hall Montreal for the next lecture. I think that's starting from 3 o'clock. So you have just a couple of minutes. Is there any question? Yes.
Thank you very much for excellent presentation.
Thank you.
What you did by AFM, you tried to catch the mechanical characteristics in one direction by AFM. You didn't do any LFM to catch the mechanical properties in lateral directions. To understand the mobilization, it is necessary to do also the LFM. The AFM is not enough. That is just the comment, not question. You will catch the mechanical characteristics in lateral direction by LFM.
Not only lateral, height. Lateral is not very well resolved by IFM, but the height is well resolved.
Yes.
Very high precision. And if alpha synuclein packed in there,
Yes.
start getting out each other, it will make substantial height changes. Then when the fibrils are formed, the height changes more. These are very small patches. Can be 100 nanometers in size to 1 micron on a very flat surface. Flatness can be better than 1 angstrom, 10 to a nanometer. Therefore, you know what's going on there. Moreover, the resolution of these instruments has changed tremendously in the last few years. You can actually resolve molecule by molecule. Have I answered your question?
Okay. Is there any other question? If not, I would like to thank all the speakers again.
Thank you.